minute break after this next presentation. So with that, let me hand this over to you, Rachel.
Okay. Thanks, Rick. Real quick, can you see, you can't see the presenter version, right? You can just see the one slide.
We can see the presentation version. So yeah, you have full, it's on full screen right
Okay,
now.
good. All right. I've got two screens here, so I want to make sure you all are looking at the right one, and hopefully you're not screaming for coffee after this presentation, but I definitely understand the need for a pick-me-up mid-afternoon. So again, I'm the Extension Vegetable Specialist. I'm based out of Lexington with statewide responsibilities. So this presentation will be kind of vegetable focused, but I think it's still going to be fairly useful or relevant for anyone who's curious about root knot nematodes. So just real quickly, nematodes in general, so not all nematodes are bad. I'm going to be talking about you know, the harmful ones to plants, but actually that represents a very small percent. Plant parasites are about 7% of all the nematodes out there. The others are fresh, marine and freshwater. There's soil, there's invertebrate parasites. So there are human, you know, parasitic nematodes. But I will be focusing on the plant parasitic ones. So for those of you less familiar with nematodes, they are non-segmented roundworms. So they're not like earthworms. They have cuticle annulations and bilateral symmetry, just like humans. When you look at them under a microscope, they don't look like that. We're kind of seeing them on their side, but if in reality they have that split down the middle, just like us. No, there's no respiratory or circulatory systems. They get their oxygen by diffusion, and they move dorsal ventrally. So they don't, plant parasitic nematodes don't move that much in the grand scheme of things by themselves, but when they do move, they move like a dragon, like up and down like this, rolling, versus a snake, which would kind of move side to side. So just to give you an idea on that. Nematode reproduction, there's 3 options: hermaphroditic, sexual, and parthenogenic, which is kind of a version of asexual reproduction. And the reason I bring that up is that will become relevant in a later slide. So that's basically, there are males and females present, but the males aren't necessarily required to produce eggs and offspring. So the basic nematode life cycle is an egg, there's an adult, right? Wherever I want to start, it's circular, egg, and then there's a juvenile that's still kind of encased in a clear kind of egg sac. And then J2 is the first kind of vermiform, worm-like looking juvenile, J3, J4, and then they become an adult. So specific to plant parasitic nematodes, if you're wondering why is this important, they can cause billions of dollars worth of damage worldwide. This number is likely an underestimate, and it may be out of date. A lot of it does happen in, you know, the tropical regions, but here in the US, we do $6 billion is a significant amount of money. Soybean particularly, and so it can affect the agriculture industry for sure. And what makes them a parasite? Well, they feed on plant tissue. All plant parasitic nematodes have what we call a stylet, which is a protrusible spear. So this would be the stylet. This is the stylet, you know, protruding out. Normally, it would just look like that. When you see it under a microscope, typically it's not sticking out. So they can be found, plant parasitic nematodes can be found on the outside of roots, on the inside of roots. They can stay there, and I'll focus on these in particular for the rest of the presentation. They can move around, move in and out of roots, and they can be found, in some rare cases, be found on foliage. So how do they cause damage? Direct damage just from feeding, right? Removing host tissue, They can alter host physiology. They can allow for secondary infection by another organism. So similar to that, transmitting other pathogens, so they can be a vector for a virus, for example. They can interact with other pathogens in a synergistic way. So similar to this secondary infection, they can create wounds on roots, and then another organism, another pathogen can move in. And they can increase the susceptibility of their host plant to environmental stress. And that's a big factor. So I'm going to be focusing on Meloidogyne species, which is also known as root knot nematode. It is a sedentary endoparasite. There's over 100 species, but 4 species, roughly 4 species account for, more than 50% of what is found in the US crops. So Meloidogyne incognita, that's known as southern root knot nematode. Meloidogyne hapla is northern root knot nematode. Meloidogyne javanica, that's Javanese root knot nematode, and Meloidogyne arenaria is peanut root knot nematode. So the females can produce about 500 to 1,000, sometimes up to 1,000 eggs at one time. They do not look like worms. The females are globular in shape, and you can actually see them with the naked eye on the outside of roots sometimes. So the vermiform, the juveniles and the males, those cannot be seen typically with the naked eye. Those would not be able to be seen with the naked eye, but the females are big enough that you can see them on the roots. They're usually, it's usually said that they prefer sandy soils, but as I will kind of go into a little bit, they can still be pretty much found a lot of places, regardless of how sandy the soil is. And so you may be asking yourself again, why is this, why do I need to know this, or why are we talking about this? Well, my lab recently, in the last 2 years, has conducted a survey across Kentucky of root knot nematode in high tunnels. And so we focus on high tunnels because of often the lack of rotation and the high soil temperatures. And so we thought those kind of conditions would be conducive for root knot nematode. So a constant host is available, right? Usually more intensive cropping in a high tunnel. Often it's the same kind of plant family, maybe even the exact same crop is there year after year, season after season. And perhaps not very much sanitation. And, one of the benefits of high tunnels is this increased soil and air temperatures. Well, in this case, that may be driving a little bit of increased nematode presence and reproduction. So we were able to survey 62 counties, and that included 175 high tunnels. There were 78 high tunnels where we observed no root knot nematode. So we collected soil samples and then sent those to a lab to extract the root knot nematode, if it was there, from those samples. So 78 samples had no root knot nematode observed. 97 had root knot nematode observed. And of those 97, 53 were identified as southern root knot nematode, otherwise known as Meloidogyne incognita. Meloidogyne hapla, or northern root knot nematode, were in 38 of them. Peanut root knot nematode was in 4. One sample is still inconclusive, we think, because the DNA was just poor quality. And you may be doing the math on this and wondering, okay, you're missing one, and that is because one high tunnel had both Meloidogyne hapla and incognita. And so I found that to be fairly interesting. But you can see here, blue is no presence of root knot nematode. The yellow star is southern root knot nematode. Hapla, or northern root knot nematode, is this orange or red diamond. The little purple trapezoidy thing is the peanut root knot nematode in the one inconclusive sample. So they're everywhere. They're all over the place. And from my eyes, I cannot see a clear distribution. So southern is up northern Kentucky, Northern is in Southern Kentucky, so it's certainly not, you know, divided by any kind of latitudinal line. So again, this is the female with the egg mass. This is a female root knot nematode. This is a vermiform nematode, and you can see the stylet here. So the life cycle specifically for root knot nematodes is really, okay, we could start with a root knot nematode female with an egg mass, right? And then those eggs hatch, and the J2 is the infective stage. So those are these juveniles. The second stage juvenile is what penetrates roots, plant roots. They usually overwinter as eggs, but the juvenile will then turn to J3, J4, and then an adult, either a male or a female, and start the cycle over again. And the complete life cycle can happen in as little as 2 weeks, up to 30 days. It really depends on the conditions. Specifically, the soil temperature can be really influential in how quickly the root knot nematode reproduces. So damage, right? So when we're looking at damage, we're looking at galled roots, hence root knot nematode, right? That's where it gets its name. The galling is not necessarily equal to reproduction. Those are two different kind of measures, but it's certainly indicative of the presence of root knot nematode. And usually severe galling indicates a higher population density of root knot nematode, but different species of root knot nematode will have a different influence on the severity of the galling. So here's probably a tomato root. This is carrots and sweet potato. So obviously, right, these would be unmarketable. This would likely be unmarketable, right? Tomato roots, right? We're not eating the root part, but this, and as I'll show you, could really influence the above ground development and yield of the plant. So again, here's a close-up of that. So symptoms and damage. So, right, this is pretty obvious, but you know, if you're not pulling up the roots and really looking, you may not see this. And this is severe. This is severe galling. This is a non-resistant plant with severe galling. And so I think most people would notice that something's not right here, even if they didn't quite know what they were looking at. But something like this, so this is in a trial where we had confirmed southern root knot nematode, and these little burned leaves down the older leaves here, this is the beginning of those symptoms, which I think a lot of people would not necessarily chalk up to, oh, I've got a root knot nematode problem, right? This could be nutrient deficiency or perhaps even drought stress. And so that, I think, is why people have issues with, you know, recognizing the problems. And then, of course, once you get to this stage, right, you know there's a problem, but even at this stage, you may not know it's root knot nematode. There could also be other pathogens involved, leaf mold, early blight, right? There could be a lot of other things happening at this stage of the summer in a high tunnel. Okay, so management options, right? So prevention. Do not move the nematode, right? Do not bring it into your farm. That's ideal, right? Sanitize equipment. If you're going to share equipment, you know, if you're borrowing someone else's piece of equipment, clean it before you use it. If someone's borrowed your equipment, clean it after they use it, right? Use certified clean planting stock. You can do resistance with the MI gene. I'll go into that a little bit. I'll go into that more. There's usually, actually it is for M. incognita and Arenaria, and I think Javanica, there is resistance. But for our purposes, mostly M. incognita, southern root knot nematode is what's going to be most relevant for us. Cultural, that would be something like soil solarization, Biological, like antagonistic fungi and bacteria. Chemical would be fumigants and nematicides. So factors that affect efficacy, right? The application timing, right? Are you targeting it when it's going to be most effective, when the populations are perhaps active? Repeated applications, the mode of action, the movement in the soil. Are you getting, if you're applying a chemical, are you getting through the soil profile, for example? Are you targeting the nematode appropriately? The soil type, the soil moisture, the soil depth, right? All of those things are going to affect efficacy. So we'll delve a little bit deeper into prevention, right? So the goal is to keep plant parasitic nematodes out of the soil and or keep the populations very, very low, right? And so this could be achieved by practicing regular sanitation, using clean planting stock, practicing non-host crop rotation. It does have a really wide host range. So that's going to be something, you know, you can follow up with me or talk to your county agent, or I have a fact sheet that I can show you at the end of this presentation. Those kind of things are going to matter. Maintaining soil health and quality, inspecting plant roots, tracking yield can really affect, you know, if from one year to the next, you may not notice it, but over the course of a few years, you may see your yield declining. And it can kind of sneak up on you if you're not tracking it. And also, of course, testing your soil. So getting it tested, getting it evaluated for root knot nematode would obviously help you in determining whether you've got a problem and trying to stop it or manage it. Cultural management would be to improve soil health and quality, enhancement of the soil's ability to suppress plant parasitic nematodes, minimize population buildup, enhance crop resilience. That can be achieved by water and nutrient management, cover cropping, non-host crop rotations, solarization, for example. So we did a study last summer looking at solarization in a high tunnel with root knot, or 2 high tunnels with root knot nematode, Meloidegon incognita. And as you can see, the non-solarized control had much higher soil populations of root knot nematode compared to 2, 4, or 6 weeks worth of soil solarization. In the tomato roots, so we planted after solarization, tomato was planted, and again, non-solarized. So this is after several months of growing a tomato plant. The non-solarized control had much higher populations. The plants that were in that area that never received any solarization had much higher populations of a root knot nematode eggs in their roots compared to 2, 4, or 6 weeks worth of solarization. Biological management, so biofumigation, that's an option. This could be categorized as chemical, but it is typically you're using a plant. So it's naturally, you know, a chemical is naturally produced from typically brassicaceous crops like mustards. It can be supplied in seed meals. It's commercially available in the product Dominus, and it's allelopathic. So plants with this chemical, allyl isothiocyanate, are allelopathic, and they can negatively affect nematodes. That's helpful, but also other plants. So you just want to be careful if you were going to use that. Antagonistic fungi like arbuscular mycorrhizal fungi, those would also be considered under biological management. Chemical management. So this is not for Kentucky, for vegetable growers. It's not it's really expensive. It usually requires a commercial applicator, and in high tunnels, it's not really permitted. So it's not a terribly useful strategy, in my opinion, for Kentucky vegetable growers, but it is an option if someone, you know, for larger scale farms, it could be the option. So there is the component of beware of the boomerang effect, which is, you know, you've suppressed something and then it bounces. If you don't get it all, which often doesn't happen, beware of that boomerang effect. Resistance, the goal would be to reduce the population density by introducing a non-host. reduce damage to the crop and yield. And so that can be achieved by selecting plants that are genetically resistant to the problem nematode, grafting resistant rootstock. And so caution, right? Resistance must be matched to the targeted nematode species. It can be broken, resistance can be broken under stressful conditions like high temperatures. 28 degrees Celsius is about that. So that's, you know, fairly hot in a high tunnel. That's in the 90s, I believe, but just be aware of that. And tolerance is not the same as resistance. So a tolerant crop may persist and have decent yield, but the nematode populations will not necessarily decrease. So those two words are not necessarily interchangeable. So this is marketable tomato yield from evaluating grafted resistant plants to a non-grafted control. So this is tomato. We did this 2 years, and you can see the yield in 3 of the 4 rootstocks, plants with grafted rootstocks, performed better, significantly better in both years compared to the non-grafted control. And we had lower populations in the grafted treatments compared to the non-grafted control in both years. So integrated management is really crucial here. So when multiple methods are used, the chance for successful management is much higher. And we saw that when we combined soil solarization and grafting. So here, the non-solarized, non-grafted, non-resistant treatment here had much higher root knot nematode populations compared to anything else, right? So if it was non-grafted, non-resistant, but it was in solarization, that had lower populations. If it was resistant, like these 3, resistant but non-solarized, it also had non-grafted or had lower populations. So combining the 2 really helped. And again, we saw the same thing in tomato roots, the eggs that we found in the roots. So definitely non-resistant was a big influence, but resistant and combined with soil solarization, that had a big influence on efficacy.
2 minutes.
So, again, you take one of those puzzle pieces away and you lose part of your efficacy. So integrated management, from my mind, in vegetable crops, right? You need to know what nematode you're dealing with. You need to know, if you're going to use, for example, a non-host cover crop, right? Make sure it's not a host. When the crops are stressed by some other factor, nematodes can cause greater damage. So proper crop nutrition and soil fertility are really important to this puzzle, this IPM puzzle. And so make sure that that is not, the factor drawing away from success. So Archean management, root knot nematode management will require more information about nematode biology, and there isn't really a one size fits all. But multiple plant parasitic nematode species also may occur in a field, not just root knot nematode. And so understanding that component and the balancing act that may need would be important, and using a combination of management practices is required. So here are IPM or ID 36 has some information on plant parasitic nematodes, really generally, chemical fumigation and nematicides. And then this fact sheet has more in-depth information on root knot nematodes specific to vegetable cropping systems.